Sir, good afternoon.
Good afternoon. Can you
Good
hear
afternoon.
me?
Yes, sir. You're audible. Good afternoon,
Good.
everyone. A very warm welcome to this plenary session of National E-Conference on Metabolomics. I am Dr. Lalit Rathi. I have the pleasure of serving as a moderator for this session. This conference brings together experts and emerging voices in metabolomics to share their insights, innovations, and latest research. This plenary session will explore a highly specialized and impactful area within microbial biotechnology and metabolomics. It's my honor to introduce our distinguished speaker, Dr. Kanhaiya Kumar. Dr. Kumar is the senior scientist in the fermentation and microbial biotechnology division of CSIR, Indian Institute of Integrative Medicine, Jammu, and an honorary assistant professor. He received his PhD in 2014 from the Department of Biotechnology, Indian Institute of Technology, Kharagpur. Previously, Dr. Kumar has contributed to the research as a senior scientist at Advanced Biomass and R&D Center, KIST, South Korea, and as a postdoc at the Norwegian University of Science and Technology, Norway. In the early part of his research career, his core research area was bioenergy production, secondary metabolites extraction, and carbon dioxide sequestration using microalgae. Later, while working as a postdoc at Norway, his research domain expanded to the deep phenotype study of various microorganisms, especially for antibiotics production from the streptomyces, using tools and techniques of metabolomics and fermentation technology. He is currently involved in identifying, extracting, and mass producing novel bioactive biomolecules. He also has several years of teaching experience in biochemical engineering and allied subjects to national and international students during his brief stay at the uh, National Institute of Technology, Calicut, NTNU, Norway, and CSIR IIM, Jammu. Dr. Kumar has published around 25 scientific articles in peer-reviewed journals of international repute and delivered around 30 scientific talks at national and international conferences. He has around 2,300 Google Scholar citations with an H index of 16. He served as a guest associate editor in the reputed journal Frontiers in Bioengineering and Biotechnology. Dr. Kumar has been the recipient of several Indian and foreign grants and fellowship, namely the Federation of European Microbiological Societies Congress Attendance Grantee 2019, a foreign travel grant by DST and DBT, travel grant from Digital Life Norway Research School, and a senior research fellowship by CSIR. He has been associated with several reputed scientific societies such as Norwegian Society of Microbiology, the Biotech Research Society India, Asian Federation of Biotechnology, European Federation of Biotechnology, Digital Life Norway, the Korean Society for Biotechnology and Bioengineering, South Korea. Today, such a giant researcher is with us, and he will be presenting on the topic a bioprocess metabolic investigation of heterologous antibiotic gene cluster expression in Streptomyces superhost strain. This work lies at the intersection of synthetic biology, microbial engineering, and system metabolomics, offering insight into optimizing antibiotic production using host engineering strategies. So you are most welcome, sir. Before we begin, I request to all the audience, please keep your microphones muted during the talk. And if you have the questions, please feel free to type them into the chat box. We will address them during the question and answer session following the presentation. And now, without further delay, I invite Dr. Kumar to take us through his presentation. So, Dr. Kumar, the virtual stage is now yours.
Thank you so much, Dr. Lalit Rathi. I am thankful to Dr. Rajendra Ganjiwale, Dr. Manisha Purani for giving such a wonderful opportunity to present some of my work in this platform. My slide is visible, if I am correct.
Yes, sir, visible.
Yes.
You are audible too.
The topic of my today's presentation is a bioprocess and metabolomic investigation of heterologous antibiotic gene cluster expression in a Streptomyces species superhost strain. So first thing is that why this study is important. So there is a need for novel natural product discovery. And why we urgently need a novel natural product discovery? Because antibiotic resistance of pathogenic bacteria are increasing. And then we have a limited efforts to develop antibiotics. Limited efforts to develop new antibiotics. And then many times what happens that when we explore the nature to find out some antibiotic, then what happens that we rediscover the same antibiotic. So there is a high rate of rediscovery. And then we know that there are several diseases. Still, we have several diseases which are non-curable in nature. And then we need to have some new drugs. So we need to have something where we can efficiently synthesize new drugs. And as you can see in the slide, there are how resistant bacteria is increasing. In the first picture, you can see that how mostly in the ICU where we use lots of antibiotic, like in the US hospital, the vancomycin resistant bacteria pathogens are increasing. And then there are several microbes which are getting resistant to several of the known antibiotics. So keeping this thing in mind, this is one definitely this is certain that we need to have some method where we can discover the novel natural product. So this work is mostly done in the Norway during my last tenure as a postdoc. And the project in which I was working, that project was called In Biofarm project. In Biofarm, so what is the major motto of this project? To have a genetic platform, which I just said in the previous slide, that to have a genetic platform to create a superhost strain for the heterologous expression of gene clusters associated with secondary metabolism from other streptomyces as well as organisms from other genera. So when we say the superhost strain, it means that a strain, a clear strain which has genetic potential to produce large amount of primary precursor is primary metabolites, and which can synthesize the secondary metabolites. So we need to have a platform. The goal was to have a genetic platform. It will be just like that we have a blackboard where something is written, and then you just want to clean back blackboard, so that you can express any gene cluster, whether it is from a streptomyces of the different organisms or from the streptomyces itself. So either it is heterologous expression or homologous expression. And again, why we need to do this thing? Because high rate of scaffold rediscovery from cultivable organisms at currently 99.9%. So most of the time it is scaffold is rediscovered. And then what happens that the natural host strain, if you look into the natural host strain, then what you'll find that there are several silent gene clusters. So gene clusters are there, but those gene clusters are not being expressed in its host strain. So we need to deal with that. How to express those silent gene cluster in a place, in a host strain, where it can efficiently phenotypically express. Then another problem is that there is low or almost no production based on the express gene cluster in native and heterologous host. So you just think about that. We have a host, but gene cluster is also there. But even if this gene cluster is also expressing now antibiotic gene cluster, let's say here, but it can be anything biopharmaceutically important molecule, but its production is very low or too low to even detect through the advanced analytical techniques like mass spec. And then what happens that we need to have bioactive discovery approaches that are not well integrated, often relying on one technology. So we need to combine the separate interdisciplinary, we need to go through the interdisciplinary approach. So this was the main theme, main reason, rationale, because of which this project was going on. So in this project, if you can see that there are several interdisciplinary domains: biotechnology, molecular biology, cell biology, robotics, then high throughput scanning, microbial physiology, fermentation, downstream processing, omics analysis, chemical and biochemical analysis, mathematics, modeling. So several disciplines were integrated here. Different groups were involved. 3-4 groups were from Norway, and all the from the European Union. Several groups were involved in this project. They have defined objectives. But our work was mostly on the microbial physiology, fermentation, and omics analysis. So metabolomics kind of stuff. So my lab was is a very advanced lab in the field of metabolomics. And we are mostly dealing with the primary metabolites. And the strain of my model system in which we were working on, which was the main model system for this project, was Streptomyces silicolor. Why Streptomyces silicolor? Because most of the bio-origin antibiotics are produced by Streptomyces. And then this antibiotics are gram aerobic, gram positive, and it dwells or it inhibits in the soil and having filamentous growth. You might feel it when the first raindrop falls in the soil or earth. Then we smell some nice. We have a nice smell coming from the earth, and that smell is because of this Streptomyces. And this Streptomyces silicolor 1A32M145 is very efficient in the synthesis of polypeptide precursor, acetyl-CoA. From several decades, 2-3 decades at least, people are working on this Streptomyces silicolor. And so Streptomyces silicolor M145 is a model strain often used to investigate the regulation of antibiotic production. It is considered as a model strain for antibiotic production. And its genome is also sequenced and annotated among Streptomyces species. So before I speak further, it is very important to understand that because each slide is related to previous slide, so we need to be careful. We need to understand or be attentive in the lecture, in this talk. So as I just said, that Streptomyces silicolor, we call it as wild type. And there were several groups which were involved, mostly this Gomez and Marvin group, which is in London. And their main goal was to use synthetic or molecular biology to create a superhost strain. And superhost strain means the strain which have a less metabolic burden. They don't have much metabolic burden and a clean metabolic profile, and efficient in the synthesis of any antibiotic if it is introduced. So you can see the example from here that Streptomyces silicolor we consider as a wild type here. Then what happened that this group, what they did, that this Streptomyces M145 has a four major antibiotic gene cluster. But unfortunately, these antibiotic gene clusters are there, but they are not very much biologically active. So they have the precursor, they have the recipe, but it is not. Hello? So can you hear me?
Yes, sir, you are audible.
Yes. So it has four antibiotic gene clusters, and this ACT, CDA, RED, and CPK. So these major antibiotic cluster were removed from the wild type, and then it became M1146. And again, what they did that further in this four antibiotic, after removing these antibiotic gene cluster, they did some further point mutation RPOB, and then created M1152. So, I mean, there were efforts, continuous efforts to remove or to decrease the metabolic burden, so that all the energy and carbon stuff, they can be channelized towards a particular antibiotic, towards a particular antibiotic. So you have a strain, you are coming from anywhere, you have some gene cluster, you say that I have a gene cluster which has some novel antibiotic production capacity, and you introduce that gene cluster in this superhost strain, and it will start producing it. So this is by this way several mutant or derivative strains were produced. And after getting this Streptomyces silicolor M1152, now we see as empty strain because there is no antibiotic gene cluster anymore, major antibiotic gene cluster. And then they introduced one chloramphenicol gene cluster from another strain, another Streptomyces venezuela into this strain, and cross-check that whether this is working or not. So basically, in my talk, the topic of current presentation is a comparative study of empty strain or reference strain or superhost strain M1152 and its derivative, where we have introduced chloramphenicol gene cluster. So one antibiotic gene cluster has been added, and you can see in the picture that how we used to work. We used to work on spores, and then you can see the red color. One of the antibiotic is prodiginin, red color. Then this black color, actinophodin. This is also one actinophodin antibiotic. So now, now we are well aware of this fact that how we are moving in the omics system biology. In the omics world, we have first genome, then genome dealing with DNA, then we go for transcriptome, mRNA, proteins, and then we go for metabolome or metabolites, and then fluxome. So this metabolome or metabolites, these are the compound, these are the final confirmation that whether your genetic manipulation is expressing or not, phenotypically expressing or not. So this is the metabolite which is of your interest. So this is the last thing to cross-check. Again, the metabolome, if you see, metabolome can be of two types. One is the endometabolome and exometabolome. Endometabolome means the metabolites which are produced inside the cells, and some of the metabolites are excreted out the cells, which comes into the microbial media or the fermentation broth. So two kinds of metabolome. But most of the metabolomes are from the endometabolome or inside the microbial cells. You can see this picture. Picture is from SIMTEF, and how is to run several bioreactors, several fermenters to study this kind of work. And it is very important that these metabolomics, metabolites are very sensitive. So working with these metabolites are not that easy. It is very challenging. And then our goal is to correlate. Our goal is to represent that whatever the sample we are taking from the culture, that should be the representative. So since metabolites, the turnover of these metabolites are very high, metabolites are very unstable or thermal level, it can degrade very easily. So we need to have develop the method, sampling method. It is very important. It is very important to have a very efficient sampling method. So this is how we used to do. So before I go for metabolomics, I'll spend some of time in the bioprocess side. You can see at the lower side we have three pictures how in the bioreactor we used to run the this fermentation, and during the growth phase they shift from one antibiotic to another antibiotics. This can be visible from here. We used to have a media, and that media from the previous work itself, we used to have somehow two carbon source. We know that in the media we put carbon source, then only it will grow. But in this media, though it was synthetic media, we used to have two carbon source. One was glucose, definitely, and then we used to have glutamate. And it has been well established fact now that nitrogen limitation and phosphate limitation to the microbial culture induces antibiotic production. So in this work, what we used to do that we used to deprive from nitrogen source or phosphate one. In the left panel, you can see that we used to do glutamate limitation. So we used to design media in such a way that glutamate will be consumed after some point of time. And we know that this glutamate was a source of carbon as well as nitrogen. In the right panel, you can see that we used to design a media in such a way that phosphate, which is one of the component of the media, get exhausted after some point of time. And why the consumption of these two metabolites in the separate experiment were required? Because as I said that its consumption is important to trigger antibiotic production. Now you can see in the left panel that this left panel top figure A, that this is the CO2 evolution in O2 consumption. So when microbes grow, so they consume oxygen from the environment. So there will be consumption of oxygen, and then they release CO2. So CO2 will evolve, and we can correlate that CO2 evolution is more or less proportional or correlated with the microbial growth. So as you can see in the figure A, that when you have a glutamate limitation, there is a sharp decline in the CO2 evolution and oxygen consumption profile. And same goes for the DO depletion. And the top, in the similar way, in the phosphate limitation, you have a decline in the CO2 evolution and oxygen consumption. And in the bottom panel, figure B and D, they are the offline data. Offline data means we did the assay. And here you can see that both A and B are correlating to each other. When there is a decline in CO2, then when there is a microbial growth is inhibited. At the same point, glutamate is consumed completely. So these data are very much well correlated. And when glutamate was completely consumed, we have antibiotic production, which is visible in both figure B and figure D. So take-home message from this slide is that this L-glutamate, which is a nitrogen source, and phosphate, two are the major important compound. The deprivation or depletion of these compounds are essential to trigger antibiotics, and they are correlating with each other with online data and offline data. Now, similar kind of result we got with compared to M1152, which I just said in the previous slide, that M1152 is an empty strain and M1581 is the microbes where chloramphenicol gene cluster is introduced. And similar kind of graph you can see figure A and C, the online data. Online data means it shows how much CO2 is getting evolved and how much oxygen is consumed. And the bottom figures B and D, they are the offline data. Interestingly, from this figure, you can see, if you see figure A and B, which is for M1152 empty strain, you can see that there is no product formation. There is no antibiotic synthesis at all. But in panel C and D, figure C and D, you can see that there is a chloramphenicol production, which is represented in cyan color. So in one side, left side, you have an empty strain, which reference strain, no antibiotic production. In the another panel, you have a same strain, but now there is an antibiotic gene cluster. So it is very interesting to see that how these two microbes are behaving in terms of bioprocess engineering. And then you can see that there is a in figure A, you can see that CO2 is declined at 2.1 phosphate limitation and nitrogen limitation. Both happened simultaneously. So from this perspective, what we have observed that a high specific growth rate, CO2 evolution, and glutamate consumption rate was observed in the reference strain, Streptomyces silicolor M1152, as compared to Streptomyces silicolor M1581, and this shows a simpler metabolic burden in the reference strain. So, as I said previously, that superhost strain means it should have a simpler metabolic burden. And this is visible from a bioprocess perspective that no antibiotic is produced in the reference strain, and then the metabolites are consumed in a much faster way. Here, glutamate, the rate of it has a high specific growth rate. Why? Because it has a simpler metabolic burden. It has a higher CO2 evolution rate. Why? Because it has a simpler metabolic burden. And then it has a glutamate consumption rate, high glutamate consumption rate, because it is consuming and distributing to biomass production. Now coming to this was bioprocess side, from which we have we try to find the differentiate between these two microbes, these two strain, Streptomyces, one reference strain and another chloramphenicol gene cluster expressing antibiotic producing strain M1581. Now we are coming to metabolomics. In the metabolomics, here you can see that, as I just said, that sampling is very important. When you are dealing with the microbial culture, our goal is to see that your metabolites should be representative of what is happening at the bioprocess level. But what happens in general, when you run the fermenter, then you take the sample and then you process the sample, because of which what happens that it takes lots of time. Sometimes it's quite significant, even if it takes few minutes. It is very important because metabolites, the turnover of metabolites are very high. It can degrade within a minute. So it is very important to deal with this kind of sophisticatedly deal with the sampling process. So what kind of sampling we used to do? First, we used to cultivate the microbes, and then we used to quench the samples. Quenching sample means we used to snap freezing. We wanted to ensure that whatever the if there is a CO2 drop, then it should be completely representative to that point of time. So quenching of the sample. And then we used to sample processing, different processing we used to do. And then in the method, eventually we have seen that we need to follow two kinds of method, whether you derivatize the metabolites or without the derivatized metabolite. So derivatization we used to do. And after that, then you have a sample. Now you have to analyze the sample, and when you are analyzing the sample, then first you need to go through to choose which separation technique you need to take, LC, GC, something like that. Then in the mass spec, you need to see that what kind of ionization is required, and then its detection, whether you want to go for quantitative or qualitative. So this is the simpler, very simple way of telling that what was the sampling process of the sample from the microbial sample. So here you can see that this analysis, there can be two kind of analysis, one non-targeted analysis and another is the targeted analysis. So non-targeted means it is more like it is not absolute quantification, it is a more like normal relative abundance. And in the targeted analysis, what we used to do, that we need to have absolute quantification of the metabolites. And there are only very few labs in the world which are dealing with the absolute quantification of the metabolites. And we were mostly focused on the primary metabolites, so central carbon metabolites. But even telling to these methods and the workflow, there are several compromises. Compromises are like no single analysis or analytical instrument can cover the whole. Hello?
Yes, sir, you are audible. Please share your slides.
It is disabled, I think, first.
Yes, you are now co-host, sir. Yes.
Is it okay? Is it visible?
Yes, it's visible, sir. Make a
Okay.
full screen, please.
So I was telling about the challenges. No single analysis or analytical instrument can cover the whole metabolome. And then we have a challenges in no standard approaches are there. And then heterogeneous classes of compounds, as I said, that different physiochemical properties are there. And then many low abundant and some very high abundant. So metabolite, if you see, some metabolites are found in a very trace amount and some metabolites are found in a very high amount. So you have to deal with all kind of metabolites. So it is not that easy to quantify these metabolites. And as I said, that main goal of my work was to target analysis. So mostly the central carbon metabolome. Now here you can see this is in the nutshell what we were doing. So this we call as a omics plot, and this is the central carbon metabolome, and it represents glycolytic pathway, PPP pentose phosphate, and TCA cycle, and then nucleoside phosphate, amino acids. These are the metabolites. So we used to try to cover more than 120 metabolites, primary metabolites, and these are the metabolites. It is very nicely, clearly visible here that what are the metabolites we used to cover. And our goal was to have a single method. Everybody will be interested to have a single method which can quantify or can handle all those metabolites. But as I said, that it is not easy. Eventually, we come up with the six method to quantify all 120 metabolites. So one method was capillary ion chromatography, MSMS, as I said, LC-MSMS. And then it was mostly used for the phosphorylated nucleoside or sugar phosphate. And then we have two methods. One is the PITC, PLC, and one is the OBHA method. One is used for the amino acid, another for organic acid. So there we used to derivatize it. What in the previous slide I was telling that you need to derivatize for the sum of the metabolites. And then some of the metabolites we could quantify both through capillary ion chromatography and OBHA UPLC method. And the last two, which was for NADs and CoAs, which were very difficult because these metabolites are highly unstable and very temperature sensitive. So it's very sensitive. So then we took lots of time to quantify these NAD, NADH, NADP, NADPH, CoA like CoA, acetyl CoA, malonyl CoA, succinyl CoA, all these metabolites. So in nutshell, this was our workflow of doing quantification of absolute quantification of the central carbon metabolites. And this is the mass spec. This is obvious that what we used to do. First, we used to have separated, and then we have a mass MS. First, ionization is happening that we call as a parent ion. But some of the metabolites, even they have the same M by Z ratio, but still they cannot be the unique compounds. So you need to fragment it further into the daughter ions. And then you quantify based on the daughter ions. Then only you can exactly say that this is your metabolites. You are quantifying the right metabolites. So before going further, we have one method. This method is called isotope dilution mass spectrometry method. So this method is a very, very useful for the absolute quantification of any primary metabolites. Why this is very useful? Because the biological system, when I mentioned the workflow that how is to get our samples. So biological extract, we say this biological extracts has lots of metabolite, and I mentioned that more than 2,000 or 2,500 metabolites. And the MS, when in the mass spec, when these metabolites goes, then they ionize it. But the ionization, the mass spec, even it is so much advanced instrument, but still it is not that super in ionizing all the metabolites. So some metabolites can be ionized properly, but some cannot be ionized properly. So especially when you have a sample with a heterogeneous number of compounds and a huge number of compounds. So how to deal with that? How to make sure that the ionization efficiency of your mass spec is efficient. Ionization efficiency is required because then only you can say that all the metabolites are quantified properly by the mass spec. No metabolites are left over. For this, we have come up with previously also people were doing this isotope dilution method. In the isotope dilution method, what we do that we have a sample which is 13C level. 13C level, so which is isotope, or you say a stable isotope dilution. And then you dilute your sample with that isotope, 13C isotope of the same compound. 13C isotope of the same compound. If you are able to do this thing, hello? Hello?
Hello, sir.
Yes.
Please continue, sir. You are audible.
Yes. So if you are able to dilute your real sample or standard with some we call it as internal standard. So which is 13C level. If you dilute with that compound, then what will happen? If whatever the compound which will go into even the mass spec, and as I was telling that you need to these compounds are highly unstable. So at every step, there is chances of degradation of the metabolite. Every step, there can be some degradation. So idea was that if you put some 13C labeled compound in the very beginning of your process, very beginning of your extraction process, Then if there is a degradation of the metabolite happens, then that will apply to your real sample and 13C metabolite which you have put from outside. In both the situation it can happen. So by this what will happen, that both of them will compensate it. You can track, okay, 13, so 13C will be a kind of control there. So by that you can take care of your degradation stuff. But now another question is coming, that how you will get these 13C level metabolites. Because 13C, for example, in the current study, as I said, that we used to target more than 120 metabolites. So it is very costly. It will be very costly, intensive process to buy each of the metabolites, and all are 13C level. And then there are several metabolites where you will not get this standard as well. 13C level stuff will not get, even in the commercial you will not be able to get. So how to deal with that? So approach was that here, that what if you take a reference microbes, like E. coli is very popular, and then you grow this bacteria into universally labeled 13C glucose. So all glucose, 13C level glucose, it is costly, but not that much costly. And then you grow that microbes. And if microbes grow in that compound, 13C level compound, then what will happen? Over the time, all the metabolites, and we know that it produces several metabolites, so all the metabolites will be 13C level. So eventually, in a very cheap way, very simple way, what you are getting, you can get a biological extract where all the metabolites are 13C level. All the metabolites are 13C level. So you don't need to buy individual 13C level metabolites. And then you mix both of the metabolite in the very beginning, and then you track it. That you can track it that if the reference your real sample is degrading, then 13C level will also degrade, and both will compensate to each other. And this is waters, this TQ axis we used to have in the waters. LC-MS, you have mass links and target links. We used to quantify the compound in the target links. And here you can see in the left panel, left graph, you can see that there is no linearity. There is there are three at the higher concentration. It is not linear properly, and this is the problem. This is the problem which we are trying to address. That if you have a mass spec has its own limitation. If you put a higher concentration of metabolite, then linearity will not be maintained. So solution was isotope dilution, which is internal standard we call. And in the internal standard, what we do, that then you plot a graph between response versus concentration. And here response means what? It is the ratio of your standard, external standard divided by ratio of internal standard. So this is a plot between ratio of internal standard and ratio of external standard and internal standard to concentration. And then you can compare both the panels, that you have a linearity maintained. So what is the take-home message from here? Take-home message is that if you do the isotope dilution method, and then if you use the LC-MS, then you can linearity range will increase a lot, and you can handle different level of metabolites, low and high level abundance metabolites. And this is I'm trying to show you here to the audience, because I know that in the audience now undergraduate, postgraduate, PhD teachers are there. So I'm trying to figure out very what is hands-on experience. So how this is in the target links, we used to get the data. You can see that the standard, then we used to have area of the standard, then we used to have area of internal standard, then we used to calculate response, then we used to plot it. And of course, through the software, it happens. And this is the way we used to generate the data. Now coming to our work here, you can see that this is the whatever just now I said, it was all about the workflow, metabolomics workflow, and mm, this is the central carbon metabolite. Here you can see that our goal was chloramphenicol here. It was this gene cluster which was added in the reference strain. But now let's say that you have a metabolite. We should know from the very beginning that what are the challenges in metabolomics area, metabolomic research area. One, interpretation of metabolic data is challenging as there is no direct proportionality between pool size and intracellular metabolic fluxes. Here it is important that when you do absolute quantification of metabolite, so it is the pool size, not its flux. So we are not tracking the flux here. We are just focusing on the pool size. The accumulation of in the product in a linear pathway will down-regulate the pathway flux through feedback inhibition. And then central metabolic pathway, such as glycolysis, PPP, TCA cycle, are regulated in a complex manner to both serve complete oxidation of carbon sources for maximum yield of energy and to supply carbon for macromolecules. So these metabolites can be a source of energy as well as carbon source. So it is not that easy to tell that if something is happening, it is because of what. And then microorganism in particular adapt to growth rate and metabolism in response to the cultivation condition. So this is how we used to plot our metabolite data in the heat map and different conditions, different depletion, different growth rate between the strain, and try to find out if there is some kind of correlation which we can get. So from very roughly, you can see from here that individual metabolite concentration varied up to the range of 4 order of magnitude. As I was already said that, for example, cyclic metabolite, and you have amino acids. Cyclic metabolite you will get in a trace amount, in a nanomolar, 1 or 2 nanomolar. But glutamic you can get in a micromolar, millimolar, even up to millimolar you can get some of the amino acids. So this is what we used to do, absolute quantification and representation using heat map. And this is the omics plot, which you have seen previously. But here now you can see that this is a comparative study between M1152 reference strain and 1581. And this sample we got when the phosphate depletion happened in both the cases, phosphate depletion. Now, what you can see in the left side, that all the metabolites, all the major metabolites of glycolytic pathway, PPP pathway or nucleoside phosphate, all are in the blue color. All are blue color. And even in the right panel, in figure B, also you can see that most of the metabolites in the blue color, a nucleoside phosphate or glycolytic pathway. But in the amino acid, they are in the red. So what is the take-home message from here? That when there is a phosphate depletion, then what happened? That sugar phosphometabolites or nucleoside metabolites, they all got down-regulated. They all got down-regulated when phosphate depletion happened. But in general, these amino acid, the level of this amino acid got increased in the phosphate depletion. But then question is that how come? Why this is happening? The upregulation of the most of the amino acid and down-regulation of the most of the phosphorylated metabolite was observed for the chloramphenicol M1581 strain. Okay, so why this is happening? Now, if you can correlate with the previous bioprocess data, and in bioprocess data, we know that when phosphate depletion is there in the microbial media, then what is mean that there is no more phosphate. There is no more phosphate, and if there is no more phosphate, what will happen? All the phosphorylated metabolite will not be formed. All the phosphorylated metabolite will not be formed, and this is what is happening here. All the glycolytic pathway, whether it is G6T fructose 6-phosphate or fructose 1,6-phosphate, 6-phosphate, all are phosphorylated metabolites. So there is no phosphate. So glucose will not be assimilated into the media, and these metabolites will not form. But amino acids are forming, and why amino acids are forming? That there is no more synthesis of protein synthesis are not happening. So these amino acids will be there in the media as such. It is not getting consumed. So by this way, we can correlate bioprocess data and this metabolome, central carbon metabolome data. We can correlate nicely. Now this is the another version, another advanced version of the metabolome, and this is 13C level experiment. And this experiment we did, and in this experiment you can see that the size of fermenter got reduced. Now it is only 200 ml, and you can guess why it is so, because the cost of this 13C level glucose is very high. So we wanted to cultivate in a small reactor, mini reactor, we can say. And what was our plan? And we already said in the previous slides that in our media, we used to have two carbon sources. One was glucose and one was glutamate. But now the challenge is that if I can play with the glucose in the 13C level glucose, then it can contaminate it with the carbon of glutamate. So you can see in the figure that glucose is coming from the top in the glycolytic pathway, and glutamate is going in the green from the bottom, glutamate. So we designed the experiment in such a way that glucose is completely 13C level and glutamate is completely 12C level. What does it mean? What was the rationale behind that? If we do like this, then what will happen? Any of the metabolite which is formed inside this CCM, central carbon metabolome, any of the metabolite, and if you can find out that whether this metabolite is completely leveled or not. If it is completely leveled, you can completely say that this is coming from the glucose. So this carbon is coming from the glucose, not from the glutamate. But if it is not completely leveled, then it means that some of the glutamate carbon is getting incorporated in that particular metabolite. So from this design of the experiment, we can easily say that where this glutamate and glucose carbon is going into the system. Now, before I proceed further, this is one slide I have taken from the literature, and the purpose of this slide is just to aware and to tell the audience, especially the undergraduate and postgraduate student, that what is the beauty of doing this 13C level experiment, and how they can really pinpoint or how they can really address your scientific question. For example, you can see what is written there: 1 to 13C2 glucose metabolism. So the goal of this work was that whether the carbon is going through glycolytic pathway or pentose phosphate pathway. This was the main goal. Whether carbon is going through, whether glycolytic pathway is active or pentose phosphate pathway is active. To do so, what they did, that in the glucose, they labeled first and second carbon by 13C, but 4 carbon they left it as such. This is a separate experiment from the literature, so don't compare with what is the current study. So in the glucose, you have labeled 1 and 2, and 4 are unlabeled. And then you can see that how these 2 carbon, 1 and 2, is getting incorporated at different points. So in the glyceraldehyde 3-phosphate, now 1 and 2 become 2 and 3. 2 and 3 of glyceraldehyde 3-phosphate or pyruvate. But if you come through the pentose phosphate, then you can see that in the pyruvate only third carbon is labeled. So if it is glycolytic pathway, if the pathway is followed in glycolytic pathway, then in the pyruvate you have 2 and 3 carbon will be labeled, and if it is coming through pentose phosphate pathway, then only 1 carbon will be labeled. And eventually, if you detect lactate, then you have 2 carbon doubly labeled lactate and singly labeled lactate. So if you are getting doubly labeled lactate, it means that carbon is coming from glycolytic pathway. And if it is singly labeled, you can say that it is coming from the PPP pathway. So by this way, you can tell that in your system, which pathway is active. So again, coming to our work, this is the 13C experiment. And in the 13C experiment, in the later slide, you can see that you have another how to quantify it and how to present your data. And this is called relative SFL, summed fractional leveling. So when you put 13C metabolite, 13C carbon into a particular metabolite, this is let me tell you with one example. This is PEP, PEP, phosphoenol pyruvate. This is a compound. It forms from phosphoenol pyruvate to pyruvate. So it is a 3 carbon compound. Now let's assume that you have put 13C labeled carbon into this metabolite. So it is 3 labeled carbon, it has a 3 carbon. So four isotopologies can be formed. 4 isotopologies can be formed. One will be that same PIP, which is 12C level, and this we call M+0. So there is no 13C labeling happened at all. Then there can be possible that you have a PIP in which only 1 carbon is labeled, and that we call M+1. And then it is possible that you are getting a PIP where 2 carbons are labeled, which we call M+2, and then you have a PIP where all three carbons are labeled, completely labeled, and we call it as a M+3. So if you are getting M+0, it means all the carbons are coming from glucose, all the carbons are coming from glutamate, which was completely 12C labeled. And if all the carbons are completely labeled or you are getting M+3, you can definitely say that this is coming from glucose. By this, you can have a distribution of isotopologies, and you can really tell that which from where the things are coming. But then the more easy version of this one is this summed fractional leveling, and you can see that in the there's a formula is written, and this takes care of all these isotopologies. The beauty of this SFL is to taking care of carbon distribution of all the isotopologies. So in the present study, if you have a higher SFL, it means that the more and more carbon is coming from 13C level and carbon, which is glucose in this work. And if it is less, it is from the glutamate. So by SFL, by just telling the SFL, you can say that whether it was rich with the glutamate carbon or whether it is rich by 13C level carbon. So now I'm trying to say that how we can nicely explain this one. You can see the same central carbon metabolites pool. Now 12C glutamate is going from the bottom, 13C glucose is coming from the top, through the glycolytic pathway. Now let's see that if you quantify one metabolite, let's say malate. This malate is in the downstream. So downstream of the TCA cycle. And here you can see that M0, 1, 2, 3, 4. You can see from the figure that this black color is very abundant. And black color is very abundant means what? It is molecular weight plus 0. And what does it mean? It means that it is completely coming from the glutamate carbon, 12C level glutamate carbon. If this is happening, we can definitely tell that this glutamate is following this pathway. Glutamate first is going to alpha ketoglutarate, then succinyl-CoA, succinate, funate, malate, like that. Then we took another compound from the lower glycolytic pathway, PIP, here. What you can see here that there is a black color and there is a yellow color. These two are the abundant Black color abundant means molecular weight plus 0, and yellow color abundant means molecular weight plus 3. And what information we get? If it is plus 0, it means it is completely 12C level. Completely 12C level means coming from glutamate. If it is completely leveled from 13C carbon, then it is coming from glucose, which is visible here. So it is a mixture of two. For glutamate and glucose, both it is mixture of these two carbon. So it means that in general perception is that the carbon from glucose is coming through the glycolytic pathway, then it enters into the TCA cycle, and this thing happens. And glutamate goes here only, and then is excreted. But here, what we are getting, what we are learning from here, that this glutamate is going back into the glycolytic pathway. So if we can put like this, that this is the pathway it followed. Similarly, if you see, we took another compound, top glycolytic pathway, glucose 6-phosphate, and here also you see that there are this cyan color is very much abundant, which is molecular plus 6, and it means that it is completely labeled by 13C glucose, and something you are getting in yellow color, which is molecular weight plus 3. It means that carbon from glutamate is going through TCA cycle, lower glycolytic pathway to the upper glycolytic pathway. So this is the impression which we get. Similarly, and glycolysis is already happening from up to downwards to TCA cycle. Then we also checked one metabolite from the oxidative PPP. Here it is 6-phosphogluconate. And the 6-phosphogluconate, again, you can see that one is cyan color, which is completely leveled. It means it is coming from the glucose. Yellow color is coming from the glutamate carbon. And then we have this ribulose, here molecular weight plus 2, and this color. And here also, if you carefully see the biochemistry and you track the carbon from where to where it is going, then you can definitely say that there is this is following through this transketolase, this reversible reaction is happening, and that is the reason why molecular weight plus 2 for this ribulose is forming. So, and same thing happens is happening with the citrate, and here also you can clearly tell that how this carbon is going from here. So in the citrate, if you have a citrate, how citrate is formed, oxaloacetic acetic acid and acetyl-CoA, they are formed 4 carbon compound and 2 carbon compound. Citrate is 6 carbon compound. Then what happens that if citrate is completely 2 molecular weight plus 2 means level, it means that all are 13C level. But if it is molecular weight plus 0, it means it is completely unlabeled, or we say it is completely coming from glutamate carbon. But in our case, we are getting both. So it means what is to mean that eventually at the end of the day, at the take-home message from this slide is that glutamate carbon is entering to the TCA cycle, glucose is entering into the glycolytic pathway, and all are there is a bidirectional flow of carbon. Bidirectional flow of carbon, glutamate is going through TCA cycle to glycolytic pathway, and some goes to the PPP pathway as well. And similarly, glucose is coming from the top, it is going through glycolytic pathway and then coming to TCA cycle and PPP pathway. And this was the experiment. The same thing is represented here in the stacked bar diagram and showing the difference between reference strain and this chlorophenicol gene cluster producing strain. So qualitative, the isotopology distribution and their trend for both strains were nearly similar, which shows the linkage between both the parent and derivative strain. So that is also very important to link that qualitatively both are same, look same. So it means that one is parent, one is daughter, it is confirmed. And then G6P glyceraldehyde 3-phosphate is synthesized from glutamate, feeds its carbon into PPP by reaction catalyzed by trans-aldolase and trans-ketolase. Similar thing with other metabolites, which we have seen. And then here you can see that we also did ATP, GTP, and CDP, and in the both the strain. What we see that the relative abundance of isotopology and the relative some fractional leveling in these ATP, GTP, and CDP. And you can see that it is very challenging to interpret because this ATP is 10 carbon compound. GTP is 10 carbon compound, CDP is a 9 carbon compound, and all are formed, all the fraction, all the isotopologies are formed. It's very challenging to distinguish or find out something interesting. But what you can see here, you can see this graph, this red and black color, it is for GTP and ATP in both the cases, M1152 left panel and right panel, M1152 and 1581, red and black, and both have nearly similar profile. But this blue color, it has a different profile. So we were thinking that why this is happening. And then we can easily correlate. And if you go to our biochemistry textbook, you can see that ATP and GTP are formed from the purine nucleus, and CDP are formed from the pyrimidine nucleus. And if you track the carbon of purine and pyrimidine, what you can see that the carbons are mostly from the glycine for the purine, and glycine amino acid are formed from lower glycolytic pathway, which is rich with 13C level carbon. So it means that the purine compounds, which is ATP and GTP, these compounds they are expected to have higher carbon from the glucose, so 13C level carbon. So this is the reason why you have SFL, higher amount of SFL we are getting for ATP and GTP. But in case of pyrimidine, we have mostly aspartate carbon, and aspartate is formed from OOAA. So it means what that, and we have just seen that in the malate or downstream of TCA cycle metabolite, which is mostly from the glutamate, and which is 12C level. So 12C level means the summed fractional higher 12C leveling means we have a lower summed SFL, lower summed fractional leveling. And this you can clearly differentiate between M1152 and 1581. And same thing we did with the chloramphenicol, which is our product. And you can see if we track each of these are the 6 different time points where we have seen that how different chromatogram are formed. In the first time point, you can see that the top are formed. It means top few M plus 11, 10, 9, 8, 7, 6, or maybe 5. They are also formed. So it means what? That all the 13C carbon is going into the our product initially. But as the time is passing, glutamate carbon is coming into the picture, and more leveling of the chloramphenicol is formed. And same picture is here, same the previous one, and you can see that how the lower portion is getting lower means isotopologies of less 13C carbon is getting enriched over the time. It means that there is a higher flow of glutamate carbon into the system, reflecting higher glutamate consumption rate. And this is everything is inconsistent and in correlating with our study. And this is how we see, we track the M145, the top, you can see that this was the parent ion, and there it forms our reference strain, empty strain, superhost strain, and then again we have introduced the gene cluster. So what you can see that whenever the gene cluster is there is a similar kind of profile. But when there is empty strain, you have a kind of equal amount of this 12C carbon or 13C carbon. So again, this what does it shows? It shows that a progressive enrichment of 12C level carbon into CCMs coincide with the increased glutamate consumption rate and chloramicol production rate in M1581, both the strain, especially with the Streptomyces silicular M115. The summary of the work, The glutamate limitation resulted in a sharp decline in expression and an immediate drop in cell mass concentration. This you have seen in the beginning of the slide. The simultaneous activity of glycolysis and gluconeogenesis happened. Gluconeogenesis means glucose is forming. Glycolysis means glycolysis is glucose is breaking, and both are happening simultaneously and during the co-utilization of glucose and glutamate. A significant change in the physiology of N1152 was observed as reflected through fermentation data, metabolite profiling, and 13C isotope labeling experiment. So all are telling the same picture. So same thing. So as a scientist, you feel happy that all the experiments are correlating. In streptomyces silicolor, M1152 behaved like a closed chamber because there's with low conversion of primary metabolite into secondary metabolite, and their excretion as in M1581. Biosynthetic gene cluster deleted strain had a lower and simpler genetic metabolic burden. 13C isotope labeling data shows the traces of L-glutamate carbon in all primary metabolic pathways. And PEP, one of the metabolite, seems to a mixture of PEP synthesized from 12C glutamate and 13C glucose. The relative abundance of PEP synthesized by 12C carbon was increasing during the exponential phase, and we know that PEP and E4P both are the precursor of chlorophenicol, so this again correlates each other. The last acknowledgement, the director CSIR Triple I. Then I was working in TNU, Norway. So we have a group of scientists. My supervisor was Pierre Bruhin, and then a group. And then we have collaboration with Sintef. They were working together. Sintef in Trondheim. So NTNU and Sintef, they are in the Trondheim city of Norway. And then we had several other groups across the European Union. So all we used to work together in this project, and they have a defined and assigned task. And as I said that, we are mostly interested in fermentation, bioprocess, and metabolomics. So thank you. With this, thank you.
Thank you so much, sir, for your insightful and technically rich presentation. Your work on metabolomics investigation in heterologous gene expression offers a fascinating window into the future of antibiotic biosynthesis. Thank you once again. Now, for all the delegates, the session is open to all for questions. Delegate can unmute themselves and they can ask the questions, if any. So I think there is no question from the delegates. So we will conclude the session. Sir, your session has given a deeper appreciation for how system biology and bioprocess engineering can intersect to drive innovation in metabolomics and microbial biotechnology. So on behalf of the organizing committee, I once again like to thanks Dr. Kanhaiya Kumar from Jammu. I also thanks our delegates for their active participation and all the technical team behind the scene. With this, now I conclude this plenary session. I request all the delegates to join the plenary session tomorrow sharply at 11:30 p.m. The session will be engaged by Dr. Kokre sir. So thank you once again to all.